mutated cell is strain and
Mm-hmm.
another wild type is strain, haploid
Mm-hmm.
wild type is strain, and now if we mate with if we mate those one haploid and one haploid mutated strain and one um, wild type is strain, and we will get a diploid strain. But in diploid strain, one allele is wild type and one allele is dominant negative. So the diploid strain will not grow at 37 degrees centigrade. We know, but
Mm-hmm.
in the diploid strain, a we will do transformation with a wild type gene with a wild type gene. So if the diploid transformed yeast cells grow at 37 degrees centigrade, then we can say that that was the gene which is the mutated actually.
It is again the same thing, just other way of saying what Navneet said, that you are also hoping that stoichiometrically it will overcome. What is your problem with
No, sir, I am.
Nageshwar's?
Yes, sir.
What is your problem with Nageshwar's
No, sir.
approach?
So Nageswar's approach was to transform a haploid history, but I am trying to transform a diploid history.
No,
The
no, no, no.
difference is that in the diploid,
Nageshwar's approach is fundamentally different from either one of yours.
okay,
Where
okay,
he is not he is not making the library from a wild type cell. He is making a library from the dominant negative mutant cell.
no, sir, I am. Sir, that is not the case.
Huh?
I think you're confused between me and Nageshwar.
Who?
Okay, sir.
I think Shubhajit's confused.
Yes, sir. Yes, sir.
Uh, uh. Shubhajit's answer and
No,
Navneet's answer are more or less the same. Okay. What you are saying, Shubhajit, is
okay,
that one copy will come from the haploid wild type in the diploid, and
yes,
even more will come from transformed library.
transform
Okay.
library. Yes, sir.
So that is the
Yes,
same thing, like just one extra copy is present in the east. But one can always argue that I will go for a high copy number plasmid, so it doesn't really matter whether you have 20 copies or 21 copies. Ah. Anybody else
sir.
who
I
can?
have thought about. Okay, sir.
So I have actually tried to solve the question, but like not in like I have not taken into consideration the previous steps that we came from. I just took it independently. Like suppose we have these, and then how we are going to try identifying the dominant negative mutation. So should I share the response, like the answer that?
You no, but you have to identify the gene for which the mutant is a dominant negative.
So like what like there are a couple of assumptions that I took that like for example we are assuming that it is a dominant negative mutation in a gene which is encoding for galactose metabolism, for example, and what is so like you can move on to the next person. I'll just find my
Just a
notes
minute,
and
sir.
I'll
I
come
need
back.
to take this call. Just a minute. So Akanksha, you have to start with the dominant negative strain.
Okay, sir. So then I actually solved it just taking it independently. Like this
No,
is
no,
what
no, no,
happens. This is the
no, 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 no,
mechanism.
no, 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 no,
Okay,
no, no,
okay,
no, no. It doesn't
all right.
work.
Okay.
So some other people have raised hands. I see. Ayush.
Um, yes, sir. So, according to me, the approach used by Nageshwar is perfectly fine. Um, all the steps are fundamentally right, but the thing is, as we are talking about a dominant negative mutation, which is temperature sensitive, right? So it will grow at a temperature of east, which is 23 degrees Celsius, but it will not, the strain will not grow due to mutation at 37 degrees Celsius. So one change that I would like to know, when we will transform it with a mutant strain genomic library, the wild type haploid strain, when we will transform it, we will select for the colonies that are not growing on 37 degrees Celsius but are found growing on normal 25 degrees Celsius.
Yeah.
Then those colonies that are growing on 25, we will isolate the plasmid and sequence it and see if the genes that are present, those will be responsible for the dominant negative mutation and causing the temperatures.
Yes, that is absolutely the correct answer. Nageshwar's and Ayush's approach is the correct approach. That here you have to start with the mutant strain and kind of create the dominant negative situation by transformation. Now, to give you an idea that these are the kind of questions I actually set in exams. So you have time pressure of 2 hours and you have to solve this kind of questions. Okay. So please get ready. Okay, buckle up. You will be, you will enjoy it then. Subhajit, bolo.
Sir, I have thought about another method. Can I try?
Sure.
Okay, sir. Sir, we have identified each of the genes which are involved in the in the mutation. So, if we first design primers for each of the gene and then do
How
PCR
would you
with
design the
one pair
primers?
of the,
No, 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 stop. How would you design the primers?
sir, because we know the sequence of the gene in the
How,
previous
how,
class
how,
we have
how,
we have
how?
already sequenced.
How? When did you sequence it?
So we have already identified the genes which are involved in the pathway.
No, no. You did a temperature sensitive mutagenesis screen. You found 50 mutants, of which one mutant
Yes,
did
sir.
not complement any. So
Yes,
where
sir.
did you get to know the gene sequence in this?
So the from the other complementary group, we can take the method which you have stated in the previous class.
That you will do transformation by genomic DNA library from wild
Yes,
type strains, and you
yes, yes,
so
yes.
that way you get to know all those other mutants. How you have no clue about the dominant negative one. You are assuming that the dominant
No,
negative.
I am thinking that in the dominant negative mutant, the
Mhm.
dominant negative mutation has occurred in such a in one of the gene which we have already
So that is what
identified
you
in the
are
other
assuming.
case.
That's what
But
you are
we do
assuming.
not. Yes, yes, yes.
Okay. So
But we
assumption
don't know
is very
actually in
bad.
which of the gene.
No, but assumption is very bad. You cannot assume. Let
Okay,
me tell you that these are real
okay,
problems. Like if it is a small molecule GTPase, ah, more often
yes,
than else, you will actually get dominant negative mutations, which will lock the GTP bound state. That's it. Or which will not release the GTP hydrolyzed GTP bound state.
okay,
Okay, and for them to get recessive mutation is really not easy.
okay.
Okay, so there is no reason to assume. So, like, approach completely logically. That what was the logical basis of it? That whenever you are crossing it with any strain, it is giving you the same phenotype as the dominant negative, right? So if I take the dominant negative version of the gene and put it in a wild type cell, then again it will behave like the dominant negative.
Yes.
And the only way to do that is to construct a genomic DNA library from the dominant negative. Because one of the members of the library will have the dominant negative mutant. Okay, so that's how you should approach it.
Okay.
Okay.
Sir,
Huh?
for the overexpression idea that I gave, there is actually one study which rescued dominant negative mutation of CDC34 protein.
No, no,
With,
I am not saying that it is impossible. I am saying that the other approach is more likely to give you the definitive result. Like, say you go for an overexpression study and you did not get your result, then what will you do?
then we can't do anything about it.
So that's what it is. So, like, if you put the logic for overexpression and then propose it, I will perhaps give you 7 out of 10. But if you put the dominant negative approach as, like Nageshwar or Ayush suggested, then I will give you 10 out of 10.
Okay. Yeah.
Ayush.
Sir, I did not actually understood what point Navneet was making. I mean, I did not understood the overexpression approach.
Okay. So all of you need to understand how dominant negative works. I stated it once, I am stating it again. That what is the difference between a loss of function and a dominant negative? A loss of function is simply the gene function is not there. A dominant negative is that it does not allow the wild type to function. How does it work? This will typically happen, say you are talking about a gene whose function depends on interaction between two proteins. Okay, now in a loss of function, if you do not have gene A, then gene B will not be interact with it and there will be a loss of function. If you can provide gene A from outside, you will be able to rescue it, right? But imagine that the dominant negative mutant is such that the protein structure is such that after interacting with B, the interaction is never released. So the cycling of the protein, free protein, will never happen. Or say that it is something that hydrolyzes GTP. So it hydrolyzes GTP, forms GDP, and then the GDP is not released. So every molecule will be, in the GDP bound state. Now, because of that, it will engage some of the other partners of the pathway in a dead-end complex. So what Navneet is saying, that he understands that dominant negative actually works through this kind of titration. The good way of looking at it is almost like titration. It titrates out the good proteins in the cell. So he is saying that I will supply so much of the good protein, the same protein for which the dominant negative is there, that even on top of the dead-end complexes, I will still have some good ones that will be able to form the productive complexes and take the reaction forward. That's what Navneet is saying.
So his approach is like one by one, he will overexpress all the gene in the genome. That would be really tedious, I guess.
No, it is the same approach. Like instead of genomic DNA library, you can use a cDNA library. You will understand it when I go to that chapter.
Right.
And then you can achieve overexpression. The same thing.
Okay.
Yeah.
Okay, sir.
Approach-wise, not much different.
Okay, sir.
Okay. So epistasis. Who can explain epistasis to me? There are few people who are hiding. Hoimi Banerjee, epistasis samjhao.
Good evening, sir.
Good evening.
So, sir, in a genome, what I understand from epistasis is if for a particular function, if two genes are acting, so one can be epistatic and the other can be hypostatic. Okay. So in epistasis, what happens? One gene suppresses the action of the another gene. So that is what I understand by epistasis.
You know, it is very good that you spoke in English. I still did not understand a word of it.
Sorry, sir. Am I not audible?
No. You have to give me by example.
Mm.
Goal nahi chalta hai. You need to give in concrete example that this is how it works as an epistatic partner.
Oh, okay.
Try it another time. You know, so you try it.
Okay. So, so can I take a hypothetical example, sir?
Absolutely.
So, okay, sir. So, if let's assume there are two genes, A and B. So, if B suppresses the action of A gene, so here in this case, A is the hypostatic gene and B is the epistatic gene.
But from a genetic screen cross, how would you know? Say you have two mutants. One is mutant for A and other is mutant for B. And say you know that because of action A and B, blue color is produced. Now, how would you tell which one is epistatic and which one is hypostatic?
So, sir, in that case, we have to perform some complementation analysis.
Okay, Hoimi, you have studied textbook, and that's where your problem is. You need
yes,
to think.
Sir. Yes, sir. I'll definitely try in the next.
No, I will not let you go. You have. Are you working from your laptop or from mobile?
Sir, laptop. It's my laptop.
But do you have a stylus for your laptop?
No, sir, I don't. I have a notebook.
Okay, so let me do it for you. Okay, so this is mutant A, this is mutant B, these are both haploids, okay? Now, in both the cases, say if you grow the cells in presence of X, wild type cells, you get a red color, okay? Wild type grown in presence of X gives you the red color. What will be the phenotype with mutant A? Like if you grow mutant A in presence of X, what will be the thing? Will you get red color or will you not get red color? Huh? No, no, you have muted yourself. Huh.
Yes, sir.
Huh.
So, sir, if this is the dominant, so in case of mutation, it will express.
Again, you think I wanted to discover the genes that are important for producing red color when yeast cells are grown in presence of X. So I did our standard mutagenesis screen and found two mutants
Hmm.
which do not give red color when grown in presence of X. Okay? Now I expect them to work in a pathway.
Hmm.
So this is also not going to give you red. This is also not going to give you red.
Hmm.
Now how do you do the epistasis?
Okay.
There are two options, right?
Hmm.
That first A acts, then B acts, and then a product P is produced, which in addition to X gives you red color. Or first B acts, then A acts, and then product P is produced, which in addition to X gives you red color. These are the only two possibilities, right? How would you distinguish? How would you know?
So, sir, I can think of one thing that separately we can look at the effect of each for the production of this the red phenotype that we want.
So you have it here.
Yes, sir.
Separately, you have it here.
Mm. So maybe overexpressing one, then the other in the medium.
So if you overexpress in this, only overexpression of A will give you red color. In this, only overexpression of B will give you red color. That does not help you.
Sir, I'll think about it. The experiment what we can carry out.
हाँ शौर्य बोलो.
So the interaction between the two genes, those are non-allelic, is called epistasis. And here we first to identify the intermediate product. That means first
क्या हो गया बेचारे का?
it's the first product on which the intermediate product, on which the product of gene B is acting and producing the final product. So if we mutate the gene A, next we can see the effect, and next if we mutate the gene B and next we see the effect. In this manner we can find out either gene A is acting first or gene B is acting first.
Like, give me the example from here. What other information do you need here, if at all?
So in this example, no sir, I cannot understand this question.
Okay. Someone else raised hand. I see a lot of raised hands.
Yes, sir. So can
Go
I
on.
try?
Yeah.
Sir, epistasis, as you told, that it is needed to determine the gene function, and the phenotype caused by a mutation in one gene is masked by a mutation in another gene. And for the epistasis analysis, we require two mutants that have distinguishable phenotypes. And in order to determine the order of the gene function, by testing whether the phenotype of double mutant is similar to that of mutant A and B. So here we need that the both A and B mutated in a single cell. So we can through the phenotype of that we can determine the pathway which is involved in particular uh, gene function.
फिर से बोलो.
Sir, from the starting.
Yeah.
So in
By the way,
Sir, Monica.
which Monica?
Sir, I have joined PhD course.
Okay, may I see you? Like, can you
Sir,
turn on your video?
Sir, the video is turned on, but my camera has some.
Oh, okay,
issue.
okay.
That's why.
No problem, Monica. Please go ahead.
So in epistasis, the phenotype caused by a mutation in one gene is masked by a mutation in another gene. And the epistasis analysis require that two mutants have distinguishable phenotypes. And to know the order of gene function, we need double mutant. That is mutation in both the gene A and B here.
Okay.
And and through that we test whether the phenotype of double mutant is similar to that of the mutant A and mutant B independently.
Okay. So can you do it here?
Sir, actually the example is not very clear to me. So can I try the example you mentioned in the lecture?
No. You
Hello.
have
So
to.
can I speak?
Yeah.
Yeah, sir, like in this case, if we want what will be the product if like if we have a non-mutated A and non-mutated B gene. Okay, and then like if what is the product from the non-mutated A and what is the product from the non-mutated B gene, like enzyme, and if the accumulation of first the substrate which is present there is there, then the first enzyme, like suppose A is acting first and B is acting later. So if the accumulation of the substrate is happening, then A is mutated for substrate, and then if the accumulation of the second substrate, like the product of A is happening, that means B is mutated.
But how would you know? Here you do not. You only have two mutants and a red color.
So we need the like product of in each step also. Like what is the product if A is acting and what is the product if B is acting?
Okay, so let me try to work with you. Okay, let us imagine that A is a transcription factor. Okay, fine.
Okay.
This transcription factor is absolutely necessary for production of B, which is an enzyme. Okay.
Okay.
And action of B results in production of P, the product, which when combined with X gives you the red color. It's not impossible, right? It's a fairly straightforward thing, right?
Yeah.
What will be the phenotype in mutant if this gene is mutated?
So like B will only be produced if A is non-mutated, right? So if A is mutated, then B will not produce, then red color will not appear.
Right? So A, red color is not there. What will be the phenotype if B is mutated?
Again, red color will not be there.
Right?
Yes,
very, very careful. Absolutely not complementation analysis. Okay? It's a haploid. But I have constructed a double mutant, A and B. What will be the phenotype here? Will there be red color? Will that not be red color?
Sir, yes, sir. A, B is a mutant. Small a, small b, right?
Both gene A and gene B are mutated in this haploid. In this double.
Yeah, okay.
What will be the phenotype?
It should not show red color.
Right?
Yes, sir.
Now, if you provide a plasmid containing A into this, what will happen?
Then A will produce B.
How?
So plasmid is there now.
So A will be produced.
Yeah, matlab transcription factor will be working there. But enzyme should not be there. Like the function of B will be masked.
टेक्स्ट बुक के बाहर सोचना बहुत जरूरी है. If you provide the transcription factor A, Anupam bolo. If you provide the transcription factor A from outside, what will happen? Will B be produced?
Yes, sir.
Huh? In the AB double mutant,
Yes, sir.
if I provide A, a good copy of A from outside, will B be produced?
Yes, sir.
Shreya?
Hello.
हाँ श्रेया बोलो.
Yes, sir. If we provide the wild type, like if we provide the plasmid for A, then the transcription factor will be produced. So it can basically bind to its DNA sequence, like the regulatory sequence for the transcription of B. So we will have the functional enzyme as well. So in this case,
how?
So because
Sir, B will be produced, but there is no guarantee that
So,
it
but
will
B
work.
is mutated, right?
no, sir. B will.
B is mutated. How can it possibly be produced?
So it will not be produced. Like,
Yes, sir. B is mutated.
So what will be the color?
There will be no production of
Not
red.
red.
no red
It
color.
won't, but B will be produced, right? But the final color will not be produced.
How will B be produced? The gene of B is mutated.
Mutated.
So the defective form of that enzyme will be produced.
You are assuming that there will be protein production. What if it's a nonsense mutation in the second codon? Please do not assume. Assumption is very bad. Okay, you should go by the definition that B is, this is a loss of function mutation of B. B hai nahi. Even if you provide A, no B is possible. What if you provide B?
So then we have, like if both A and B is provided to the.
Why do you need A?
Okay, acha, okay. Only if
Not
if
needed,
I'm
sir.
providing only B, then I have the functional enzyme. Then it can convert the product P into the red color.
Right, so then you will get the color.
Yeah.
So then you know the order of action.
Yes.
That B is acting downstream of A.
Downstream of A.
Who did not understand?
Sir, if only B is provided, the A is still defective, right?
But functional B is there, na? Functional B. So it is all.
You are providing the B gene product, right? You are providing the DNA for production of B. So B will be produced in the cell. A, what is the job of A? A's only job is to ensure production of B. So if you are providing B by itself, why do you need A for it? Please ask me whatever you are not understanding. Please ask me now.
We are providing the full working enzyme, or we are providing some
You are providing the DNA sequence that encodes B in a plasmid that can ensure transcription and translation of B.
Okay, then A is not.
That's how the cDNA library, genomic DNA library, etc. work, right? You ensure that they are autonomous. They will have their own machinery. I will teach those things a little later. But this is how. Bolo.
So can we not cross these with the wild type, and then we can see when A is like if we see the F2 generation, and then when we have small a, we see no effect, but like we can see that way.
Why do you need to go to small a, small a? So you want to go to a diploid.
Yes, sir.
So how is this different than the haploid A mutant?
When we have capital A, small a, but we have both small b's, and the thing is still the color is still not expressed, then we can see that. Then we can say that B is upstream of A.
Why? You have a diploid like this.
And small b, small b.
So
if we are not getting the color here, then we can say that even though A is not mutated, because like phenotypically it's not mutated, but then since B is mutated, we are not getting the color, we can say that B is upstream of A.
B is not upstream, downstream of A. Even in this example, B will be downstream of A, right? Okay. So how would you do this? You will have the A mutant, and you will have the B mutant, and you will have the wild type haploid. So you will have to first cross these to get AA. And then, no, it will be difficult. You will not be able to construct these diploids to begin with, because you have no way of knowing the genotype of these diploids. This is a little bit more complex. I cannot, like if you want, I can get into it, but it will be difficult. You will not be able to do it.
Yes,
with this kind of mutants. It is possible to do with other kinds of mutants, but not this kind. Okay.
So since B is a different gene, the one which is haploid in A, like small a, we can say that it has the capital B gene in it already.
Yes, yes, I agree with you. Then
the haploid is basically small a, capital B, and the other one is capital A, small b.
Yes.
So can we not get from there?
So when you cross them, you will get double heterozygote. When you cross them, you will get AA, BB. Then what will happen?
Then we cross two of those again.
Okay. you cross it with another AABB, then what will happen? You will get in all possible combinations, right?
Yes, sir.
How would you know which combination you have in a particular cell? Try to do it at home.
Okay, sir.
It will not be possible. और किसने हाथ उठा के रखा है? I cannot see it here.
Sir, sir, B is down regulated by A, na? Means when we are putting outside.
No. A is a transcription factor that ensures production of B.
That's fine, but until and unless we can say that double mutant of A and double mutant of B gives which phenotype, then how we are concluding that this one the downstream of A or this one the upstream of B? Means sequence, as I understand from the lecture, sequence only we can say when we can know A mutant this phenotype, B mutant this phenotype, and double is this phenotype.
No, in this case, even double is your red negative, na?
Okay, that is given.
Like as I said, that if you take an example like this, then exactly these are the phenotypes you will see. Let us go back to the one that was there in the lecture slide.
So can I ask a question?
Yeah.
Going by the example that was previously stated by, I don't know the name, creating a double, deployed and observing the phenotype, right? So what we can do is, how do I understand is, we can cross a small b mutant with a small b mutant, and we will see that it is not producing red color because the genotype of the mutant will be capital A, small b, small b.
So then you are doing exactly what I have done with the plasmid, na?
Right.
Exactly. When you are taking a double mutant,
that is similar to what we do with plasmid.
Yeah, like crossing it with small b is equivalent to providing A plasmid. Crossing it with small a is to providing it the B
small
is
B plasmid. Exactly. Yes, sir. So which of the two approaches is more laborious?
If you think of classical genetics approaches, yours will be better.
Yes, sir.
Because you do not know the sequence, yours will be better.
But in today's day and time, it must be using transformation.
Yeah.
Okay.
Because like when you have the mutants A and B, you already have the plasmids for A and B as well because you have done the library screening, right?
Yes, sir.
So it will be easier that way.
Okay.
So now let us go to these particular examples. Piyusha,
yes, sir.
Explain the first example.
Sir, here first we have A and B. So I am telling about the analysis of biosynthetic pathway. So now if first B is mutated, what will happen? We will not get the input product 3, but we will get the product 2.
Okay.
So if we see in a reverse manner, so if we get the product 2, then we can understand that mutation took place at B. Secondly, if we look at mutation in A, so if A is mutated, then neither 2 or 3 will be produced, the first product 1 will be there.
Mm-hmm.
So in that manner, we can understand that A is mutated. But now, if A and B both are mutated, then also we will not get 2 or 3 because B acts on the product of what A produce. So we will again get the first product 1. So in that case, we can understand if A, B both are mutated. In that case, A is upstream, B is downstream.
Okay. So if we just make this little easy,
okay.
Let's say I have taken a red colored chemical.
Yes.
In wild type cells, it is converted to green color.
Okay.
Okay.
Okay. Yes.
And then I have done a screen and to identify the genes involved in production of green color, I got 2 mutants in which the green color was not produced.
Okay.
One mutant is A and the other mutant is B.
Okay.
But in the B mutant, a yellow colored product was there.
Yes. Yes.
And then if we want to do it, so this way your life becomes little easier.
Okay.
Okay.
Yes, sir. So if we now look at the process, so if the wild type was there, no mutation took place, we are getting from R to G, the end product will be green. But now in our first observation, we found that our product was yellow, not green. So in that case, we understand that if we know that A and B are mutated, so that should be known, sir. So if we know that, we can understand that if yellow intermediate product is produced, that means B was mutated. So the intermediate products were not further produced green end product.
No, it's fine. So as long as you can understand, I have no problem.
Okay, sir.
Okay. So next one, Paul, are you there? Paul, are you there? Okay. Adil. Jara prakat hai ye, please. Please turn on your camera if it is possible. Adil, I cannot hear you. Umar, are you there?
Yes, sir.
Explain the next one.
Okay, sir. So here we have to analyze the order of the molecules that are involved in let's say in any signaling pathway. So here we have two proteins or transcription factor, whatever. Let's say A and P are two transcription factors, and B, and they are acting on some specific gene, some reporter gene. Let us assume that it's coding for GFP protein. Okay, so it's coding for GFP protein and green fluorescence and giving us a green color. So it's upstream. There are two kinds of transcription factor, as we can see, A and B. So B is down regulating the A transcription factor. I mean, it's suppressing the activity of A, and A and transcription factor A, it's activating, that is, it's increasing the expression of reporter gene, that is GFP. So suppose if there is a mutation in A, in transcription factor A, So, so as a result, that reporter gene will not be able to get stimulated and expressed, and we cannot have a proper expression, and we cannot have a green color. So, and this will result in a repressed expression of a reporter gene. And in other case, if we have a mutation in transcription factor B, so earlier it is, as we can see, it's down regulating the transcription factor A, but as a result of mutation, it no longer down regulate the transcription factor A. As a result, we will have a constitutive reporter expression of GFP. So we'll get a green color constitutive expression as a result of constitutive expression. And in case of, but when it comes to double mutant, and that is both A and transcription factor A as well as transcription factor B, both are mutated here. So we'll get transcription factor. Okay, so we'll get a okay. Okay, so here we'll again get a repressed expression of the reporter gene. So by this, we can interpret that, okay, that...
Fine, give me answer of this.
Okay, so again, it's the same case. Here we can see that B is upregulating, that is positively regulating the A, transcription factor A, and A is down regulating the reporter gene. But let's assume there is a mutation in transcription factor A. So A will no longer inhibit the reporter gene, and we'll get the expression, required expression of a reporter gene. Okay, sorry, let me just mutation A. Okay, repressed. Okay, so mutation in A is giving us an increased expression of reporter gene. And if we have a mutation in B, transcription factor B, so it will not be able to upregulate, stimulate the transcription factor A. As a result, A is successful in inhibiting the expression of reporter gene. So we'll again get the phenotype of decreased expression. Okay, so
what,
we'll
decreased or increased?
Sir, when mutation occurs in transcription factor B, so no more upregulation of transcription factor A. So no more. Sorry, sir. So no more upregulation of. So sir, sorry, not decrease. It will again give increase, sir. Yeah. Increased expression of reporter gene. And in case of double mutants, that is A and B both are mutated. A and B both are mutated. Okay, A and B both are mutated. Okay, so it will no longer inhibit the expression of reporter gene, and will get an increased expression of reporter gene.
Okay. Okay.
Sir?
Now,
Sir, I did not understood that last portion. So when B is positively regulating A, and A is negatively regulating the reporter gene, that portion.
Okay. Let me go back to the presentation. You should just take it logically. So B, if B is there, then the reporter will not be produced, right?
Yes.
So if B is mutated, what will happen? In this case, B, small b meaning mutated. If B is not there, what will happen? What happens if B is there?
When B is there, sir, when
just look at the screen, it should be as simple as that.
sir, you were telling about the second example.
I thought you wanted to know about this only, right?
No, sir. I was telling about that in left side. B positively.
Here?
Yes, yes, that one.
Huh. So what is the problem?
So B is positively regulating A.
Huh?
And A is negatively regulating the reporter.
Huh.
So if the mutation both happen in B and A, so what will be the effect?
Bolo? If B and A both are mutated, is A there or not there?
A is not there.
So if A is not there, then what happens?
Oh, no negative regulation.
What happens?
Constitutive expression of GFP, a reporter gene.
Khatam. That's all.
Okay.
Right?
Yes, sir.
Sir, I had a doubt.
Huh?
Sir, in the first analysis of the signaling pathway, when we from the phenotype to the interpretation, it does not logically follow. It is also a possibility that A and B are separately positively regulating the reporter, right? And so when they're double mutated, so there is.
You're talking about this one?
Yes, sir. Yes, sir.
Okay, let us construct that. Let us say both positively regulate the reporter, like B promotes expression of A and A in turn promotes the reporter.
No. B also promotes expression of reporter. It's a separate. It does not affect A at all.
No. Epistasis analysis cannot work on parallel pathways.
Oh, okay. They have to be like.
They have to be in sequence. Okay.
Yes, sir.
Fine. Done.
Yes, sir.
Okay.
So just a small
Okay.
question.
No. Hold on. I am not done yet. I am posting a question in the group, WhatsApp group. Okay. Who has not spoken at all so far? Ankit, आंसर करो सवाल का.
Sir, reading the question.
अच्छा ठीक है, पढ़ो.
So wild type diploid or haploid E cells produce a yellow color. Okay. Diploid and haploid E cell produce the yellow color when grown in presence of adenine. This production of yellow color is dependent on the transcription followed by translation of yellow Y gene. Okay. A particular mutant diploid strain does not produce a yellow color when grown in presence of adenine. Okay, a diploid is not producing so the Y gene is mutated. How would you determine if Y is being transcribed or not in this mutant? Sir, so the situation is like, the diploid cell and the haploid, the wild type diploid and the haploid is producing a yellow color in the presence of adenine. So the Y gene is functional, and the Y gene is functional only after we are getting a protein product after transcription and translation. Now in the again a mutant diploid strain, the Y gene we have to find out whether it is transcribed or not only. So particular mutant diploid strain, Y is being transcribed or not in this mutant. Y is being transcribed or not in this mutant. Yellow color when growing presence of adenine.
वो ट्रांसक्रिप्शन वाला छोड़ दो, सिर्फ बी का आंसर करो.
Okay, sir. Upon meiotic division, when 4 haploid spores are produced from this mutant strain, 2 of the 4 spores started producing the yellow color when grown in presence of adenine, while the other spores did not. What kind of mutation now? Trans element. So 2 of the 4 spores start producing the yellow color. It is not producing this yellow color in the presence of adenine, while the other spores did not. Sir, trans element.
नहीं वो छोड़ दो. First half.
First half.
हाँ वो I have not taught you the trans element part yet.
Okay, okay. So the first part means upon the meiotic division, na?
हाँ.
हाँ. So upon meiotic division, when 4 haploid spores are produced from this, so 2 of the 4 spores are producing the yellow color, means from like when the segregation is taking place from the mutant, we are getting a functional allele which is producing the yellow color. In 2 of them, a functional allele is restored which is producing the yellow color, but in other 2 that functional allele has not gone there. So what kind of mutation do you think the E strain has?
Dominant or recessive?
Sir, I think it will be a dominant, sir, because if it is recessive, then it will not come back. After segregation also, the phenotype will not come back. So it was like heterozygous dominant mutation.
Your answer is correct. Logic is wrong, so you will get 50% marks.
Yes, sir.
Think again. If it were a recessive mutation, how would it have been deployed?
There is no yellow pigment production.
So if the mutation was recessive and the diploid is heterozygous, what would be the phenotype?
If the mutation is recessive and the diploid is heterozygous, the yellow color will be produced.
So after sporulation, you are getting 2 spores producing yellow and 2 spores not producing yellow.
Yes.
And you know that the wild type produces yellow color, right?
Yes.
So certainly there are 2 that are mutants.
Yeah.
And 2 that are wild type in the
wild type. Yes.
After sporulation.
Yes.
Therefore, the diploid must be heterozygous.
Yeah. Okay. Yeah. Yes, sir. Yes, sir. If the diploid is heterozygous, then the dominant, because of the dominant allele present in 2 of the spores, it is producing a wild type phenotype. And the recessive gene is there in the other 2 spores. That is why it is not producing the yellow color. So the diploid is heterozygous. Yes.
अरे क्या बोल रहे हो पागल जैसा? फिर से सोचो. Shayan,
sir. Yes, sir.
Bolo.
The spores, 2 mutant, 2 wild type. In the
Yes.
spore, 2 yellow, 2 not yellow.
Ah.
So for if I go back, then before sporulation, it was like means wild and means A, capital A, small a, heterozygous. Should be heterozygous.
How do you know it was heterozygous?
Yeah, otherwise all the spore will be same, na? All the spore will be either mutant or the recessive.
That is the logic, that because there was a 2:2 segregation in the spores, so the diploid must be heterozygous. Now start from there.
Yeah, so diploid must be heterozygous. Now, okay, just let me see again the question. Hello. Hello.
So
Hello, sir. Yeah, hello, sir. Hello. Yeah. So diploid is heterozygous before sporulation. Now what I have to answer?
The question is whether the mutation is recessive or dominant.
Yeah. And this diploid was yellow.
No,
it was not stated. Okay. Okay. Okay, recessive, when it's diploid, recessive mutation it happens, it cannot be heterozygous, na?
Why?
Recessive homozygous, that on, no, then only it can show the particular recessive phenotype.
Language में बहुत problem है.
Okay, but you are not saying, okay, but yes, sir.
This is producing heterozygous mutation.
Monica bolo.
Yes, sir. So in the so as Sain mentioned that this diploid mutant was heterozygous. So now the mutant stain was not showing yellow color. So as we know that the mutation is dominant because it is masking the yellow color in the presence of editin, which was the characteristic of the wild type. So when we segregate, so the haploid spores were of 2 types, that is capital A, and small a, in which now now 2 will produce the yellow color and 2 will not in the presence of adenine.
ये सब तो दिया हुआ है.
Sir, yes, because the wild type, the mutant strain which is diploid, which is heterozygous, is not producing the yellow color. It shows that the mutation is dominant.
Because,
yes, sir.
One line. If the mutation was recessive, then the diploid should have been producing yellow color. The fact that despite being heterozygous, it is showing the mutant phenotype, it must mean that the mutation is dominant negative, or dominant at least.
Yes, sir.
So these are the kinds of problems that you will have to solve for your first exam. Okay. 1 hour should be enough before the viva starts. Okay, there is a saying in Hindi, Sanskrit, Maunam Shammati Lakshanam, meaning because you are not speaking, so I am taking it as yes.
No, sir, we need 2 hours.
Sir, 2 hours, sir, 2 hours,
Depends
sir,
on how
2
many
hours.
questions are there.
There will be, 4 or 5 questions like this.
Sir, 2 hours.
2 hours?
Yes.
So we
Okay,
are not
2
even
hours.
sure if 2 hours will be enough.
I'll give you 2
Sir,
hours.
more than 2 hours.
So what, is this better, or should I give you, write everything you know about recessive mutant and dominant mutant and the genetics? What is better?
No, sir, this is
This
better.
is
This is
better.
better. But we need time to analyze the questions.
Yes, you will. But for
Yes.
that, you need to study, okay? If you do
Yes.
not study, if you do not think, no matter how hard you study, it will, how many hours I give, it will not be easy for you. Because ये तो आधा है, ट्रेलर हुआ है अभी, हाँ. From Monday, like I would strongly request that you start working on your molecular biology lectures, that is the next part, right? Right after this.
Yes.
It's of cloning.
Yes, sir. Genetic engineering, yes.
So, I'll tell you that what I would eventually do in your exam, what I will like you to do. I will give a sequence and I will ask you to design primers to clone it in a vector. Okay. So therefore, because see, otherwise in this online format, I cannot ask you any question that you will find it in a textbook. Not that I used to do that ever, but now I cannot do that, period. Okay. So therefore, you need to really study hard. But the advantage is that you will know molecular biology like the back of your hand. The day your lab opens, you will start to work like you are a professional. Is that good?
Yes, sir.
Yes, sir.
Yes, sir.
कितने धीरे से सब yes बोल रहे हैं.
Yes, sir.
Yes, sir.
Yes, sir.
सबको भूख लगा है.
Sir,
Sir,
sir, as in some points, I am not understanding few things like the terms and all those things. So, how should I prepare? Where I do not know, like.
When I will eventually draft the questions, I will make sure that nothing is used in there which has not been taught.
Okay, sir.
Okay. Okay. Tada. Good night. Have a great weekend.
Good night, sir.
Monday.
Good night, sir.
Good night, sir.
Bye.
Nice.
Good night, sir.